Hi, I'm Anastasia Potts.
And I'm Gustavo Chavarria.
So we're going to talk a little bit about DNA bead cleanups, some of the challenges of doing them manually, and how a 96-channel pipetter can really help speed things up. But first, before we get to the demo, I'm going to jump into a couple of slides. So DNA purification beads, which are sometimes called SPRI beads, are paramagnetic particles that can reversibly bind DNA. So here I show kind of a typical workflow, a 0.8x post-ligation cleanup that you might use in the NGS library prep. So beads that are suspended in PEG and salt are added to your DNA samples, mixed really well, and then incubated for a couple minutes to promote DNA precipitation and bead binding. The samples are then put on a magnet to pull the beads against the wall of the tube and incubated until the solution clears. Then the supernatant, which has the unselected DNA or whatever other sample components that you want to get rid of, that supernate's taken out. Samples are then washed in ethanol with short incubations during the wash. And once ethanol is removed, the beads are left to dry for that residual ethanol to evaporate. Beads are then resuspended in a low salt buffer with lots of mixing and then incubated to allow the DNA to go back into suspension. Samples are then put on the magnet, and the supernatant that now contains all that DNA that you wanted is transferred for a new tube and is ready for whatever you have planned downstream. So bead cleanups are great, and on paper they look really short with like 20 minutes of incubation and just a few reagent additions and washing steps. But when you take a closer look, they have a lot of individual pipetting steps. For a relatively standard NGS cleanup like the workflow I just described, you need at least 27 pipetting steps for a single sample. And this just adds up. So if you're processing an entire plate with an 8-channel multi-channel, that's 324 pipetting steps. And if your workflow requires multiple cleanups, your thumb is just going to be aching and you're at risk for a repetitive strain injury or an RSI. So bead cleanups are also sensitive to pipetting technique. And this can be difficult when you're working with multi-channel pipettes specifically, just because it's a little bit more complicated and you can't always see as easily what you're doing than what you would with a single channel. So mixing and resuspension technique has a big impact on DNA yield and quality. So you can really see significant positional effects across the plate from the same user and large user-to-user differences. So pipetting speed, the tip depth, the angle that you use during mixing can all cause insufficient mixing, and it's going to vary across columns, particularly over a very long cleanup. So you can see positional effects. And as I mentioned, multi-channels can be more difficult because you can't always see every tip, and so maybe if you're shifting the multi-channel back and forth, the top of a column and the bottom of a column might be treated differently. And so those with less experience and less kind of comfort doing this with a multi-channel are also going to see more variability. And this can kind of add into user-specific effects. Now this is also even more difficult for 384 well plates where you have like no visibility into what's happening in an individual well. And technique becomes really critical because of the small volumes. I did just want to go back to the idea that there's so much pipetting here. So we have data that shows more fatigue leads to increased pipetting errors and worsens your pipetting technique. And I know this from my own experience. After a long day of pipetting, my reproducibility goes down, just because of tedious pipetting intensive workflows. And this includes bead cleanups. 96-channel pipetters are a great way to increase the speed, throughput, and reproducibility of bead cleanups and really save your thumbs. So MicroPro is a small, compact, semi-automated 96-channel pipetter. It can aspirate, dispense, mix, load, and eject tips, and all this at a press of a button rather than using your thumb. You can imagine all of the ways this will cut back on the number of pipetting steps, right? Because you have 96 channels. And it can also really shorten protocols, which we're going to show here for the bead cleanup. It can also normalize pipetting technique across columns, rows, and users. So you're really going to cut down on those positional effects and user-to-user influence on your data. You can also pre-program protocols that define mixing volume, mixing speed, and the number of repetitions, again, to kind of reduce that variability. One thing about MicroPro that's also great is that you can define the Z height, which is the height of the tips relative to the bottom of the well. And this can really have a huge positive impact on consistency in mixing, reagent addition, and you're not going to create bubbles. And you can also minimize dead volume by, making those tips go all the way to the bottom of the well. So today we're going to demonstrate a bead cleanup with MicroPro 300, which has a working volume between 5 and 300 microliters, and it's really great for 96 well plates. For 384 well plates, we would recommend the MicroPro 20, which has a smaller range between 2 and 20 microliters. So if you're running those low volume cleanups in a 384 well plate, you can still reduce the number of pipetting steps significantly and also reduce the risk of sample swaps and still get that increase in reproducibility. So now we're going to demonstrate how a DNA cleanup with MicroPro really compares to a multi-channel pipette. So for a protocol like this, where there's a lot of different steps, we would want to program ahead of time. Do you think you could give me a tour of the app and sort of how we would set up this protocol?
Yeah, absolutely. So MicroPro, as you learn, is the smallest 96-channel pipetter in the market, weighing only 11 pounds. As you can see, it is controlled wirelessly with this app. As you can see here, it has all the pipetting modes from single pipette to multi-aspirate and repeat dispense, which is very helpful. But one of the coolest modes is the custom, which is basically a deconstructed pipetting cycle. You can actually set up all the steps in any position you want, from like tip loading and tip ejection, all the way to mixing and incubating. You press step, aspirate, in this case, 100 microliters, speed 10, and then you're going to dispense the same volume and purge to complete the pipetting cycle, which is basically the second step. Now, another cool feature is how you can actually set up the Z height, which is basically how you set up the distance relative to the bottom of the plate. And it's very simple. You simply find the bottom of the well, you tap on this middle column that says none, and then create new. The app will walk you through all the steps that you need. Once I tap next, you wait a couple of seconds, and then you start tapping fine or coarse, and then the actual instrument will start moving up. Once you're happy with the distance, then you give it a name. In this case, I'm going to give it AAA. And then save. You can have the same distance to all the steps, or you can have different ones. Another cool feature about MicroPro is you can have as many distances as you need for the same plate. As Anastasia is going to actually mention, you're going to have multiple distances from the lower end of the well all the way to up high.
Yeah, so for bead cleanups, right, we're using the plate on and off a magnet. And so we actually have multiple heights saved in both those things. And in the same protocol, you can use multiple. And that's really important, right? So for an aspiration of a supernatant, right, you probably want to pull from the very bottom of the well. But when you're mixing, you probably don't want to mix at the bottom. And so this is really great for this bead cleanup workflow.
Yeah, and another cool feature about the custom mode and the Z height itself is that you can actually deactivate or activate those heights at any given time. So the distances are not bound to the actual protocol or any steps whatsoever.
Okay, well, thanks for that. And we, of course, programmed everything, so we're ready to get started. So I'm actually going to be running the MicroPro today. I'm lucky enough.
Yes.
And Gustavo.
And I'm going to run it with a Rainin XLS 8-channel pipette manually. You also see me doing it in a time lapse.
Because mine is going to be much faster.
That's right. So, shall we?
Yep, let's go.
Perfect.
Okay, so I'm going to go ahead and get my bench all set up for the protocol. So on the app, I already have everything set up. Just going to go ahead and load tips, and then my plate full of samples. Get this lid here off, and then we're ready to get started. So I'm going to go ahead and load the tips, and MicroPro takes care of loading them automatically. Just give it a second to grab. Then I'm just going to take this empty tip rack off and replace it with the beads that I've already put in this plate. And because beads settle over time, we want to go ahead and do a mix just to make sure everything's in solution before we transfer it over to our samples. Okay, we got that mix done. Just moving the beads over here. And again, this is a really important step to make sure that these are very evenly mixed, because that's really important for the bead binding and the most efficient DNA yield. So we set this up to mix at speed 9 and 10 mixes, again, just to make sure that's super well done. So now that we have that first mix done, we're just going to go ahead and let it incubate for 5 minutes. And then after that, we'll move it to the magnet. Okay, so that first 5-minute countdown is up. Now we're just going to go ahead and put the plate on the magnet. This will pull the beads to the side of the wells and allow us to remove any unwanted material that's left in the supernatant. So we just leave it for another 5 minutes. Before moving forward for the wash, we just want to make sure that the solution is completely clear and all the beads are pulled to the sides of the well, which they are. So what we're going to do is put a waste container in this third position, add a new set of tips. Just go ahead and load them. Then we're just going to remove the supernatant. And this just contains whatever's in that sample that we want to get rid of. Then we're going to go ahead and put that in the waste container, which is now just sitting underneath the deck. And we'll just go ahead and eject these tips. Replace them with some fresh ones. Just adding a reservoir with 70% ethanol. We're going to add this to our samples, and this is just going to wash the beads. This is going to incubate for 30 seconds, and we're just going to use the same tips here. Okay, we're just going to go back in and aspirate all of that ethanol. And then eject these tips. Now we're ready for our second wash. And here we're using 70% ethanol, but protocols vary somewhat, so just make sure you check your protocol. And then again, we're going to do another 30-second incubation. And you see I'm using the same tips here because it's a very short incubation. And this is actually, depending on your protocol, can be a way to save tips. Okay. I'm going to go ahead and again remove that ethanol. Add it to the waste container. Eject these tips. Replace them with a fresh batch. Now we're just going to go in one more time to our sample just to remove any residual ethanol that might be left. And then inject these tips. So removing that last little bit of ethanol helps ensure that there's basically no liquid left across any of the wells in the plate. And so during evaporation, hopefully we get that very even across all wells. And again, because we're using a 96 channel head, we know that the dry time is going to be the same across the plate regardless of column, which can help prevent ethanol carryover and make sure we get all the samples in a great place at the same time. So now we're just going to incubate for about 3 minutes, making sure you want to manually check and see that the beads are dry but not cracked. Okay, so now our beads are nicely dry but not cracked, so we're ready to go ahead and resuspend them. So first I'm just going to take my plate and remove it from the magnet, and then go ahead and load some fresh tips. Then I'm just adding my elution buffer. So I talked a lot about how important the Z height was at the beginning. And that's important for this step. So here we have kind of a medium Z height. So when I add the buffer, you don't introduce bubbles at the bottom. And when you're mixing, you get a great even mix without having any sort of accidentally hitting the bottom of the well, introducing bubbles, causing unevenness across wells. So now we are just going to have another 3-minute incubation. And during this incubation, what's happening is you're giving the DNA a chance to go back into solution and separate from the beads. Okay, so now it's been 3 minutes. We've totally released and put that DNA back into solution. But now we need to pull the beads out of solution. So I'm just going to add my magnet back, put my sample back on, and then we're going to again wait 5 minutes to make sure all the beads have a chance to go to the walls, so that we can then pull out just our DNA in solution. So now that the 5-minute incubation is up, your beads should all be against the magnet, and we should be ready to transfer our DNA in solution into a new plate. So I'm just going to go ahead and load a new fresh set of tips. Then I am going to load my purified sample plate. Now I'm going to aspirate the sample. And again, I've set the Z height here based on where the plate sits on the magnet. And pulling from the very bottom of the well to make sure we get all of that sample. And dispensing into the sample head at a slightly higher to eliminate creation of any bubbles. Just going to do a quick touch off. And now we have our nice purified samples ready to go for whatever downstream step we have. So now I'm going to welcome Gustavo back. I hope this really showed you how easy it is to run NGS bead cleanups with MicroPro, and you're going to save a lot of time, a lot of pipetting steps, and you're going to get really even yield across the whole plate.
And now we're going to continue with this webinar by showing a couple of comparison tables so you can see how MicroPro can actually expedite your NGS sample prep. The purpose of this demo is to show you the advantages of using systems like MicroPro for bead cleanups. But let's not forget there are several ways to run these protocols, like using single channels in 1.5 ml tubes or 8 or 12 channel pipettes in plates. The table on your screen are approximate times that took to run both protocols. The difference in hands-on time is quite obvious. As you can see on the table, MicroPro reduces the hands-on time 80%. And it is quite obvious. And the total time, it is 50% faster, which is quite substantial. And for my demo with 8-channel pipettes, I followed proper pipetting techniques to maximize DNA yield and consistency. But let's just stop right there for a second. Anastasia shared earlier that bead cleanup requires 324 pipetting steps. And I believe we all agree that following proper pipetting techniques for a total of 34 minutes is quite challenging, if not impossible. Keeping the pipette perpendicular at all times can be extremely difficult during the entire protocol, and it is critical in this case to avoid disturbing the bead pellet, and it's even more challenging with multi-channels. Also, aspirating from the bottom of an 8 wells is also difficult. Perhaps you didn't notice in the video, but I made mistakes and aspirated the beads while I was discarding the ethanol washes. Other techniques to consider are pipetting at steady pace or pre-rinsing that are factors that are also very difficult to maintain. In summary, it is very unlikely that your pipetting will be the same over the course of the entire protocol. The ergonomics aspects come into play as well. For example, at Rainin, we highly recommend taking breaks after 20 or 30 minutes of pipetting between pipetting steps. So remember, those incubation times are your best chance to recover and take breaks for pipetting. With MicroPro, users don't have to worry about these inconsistencies, like for example, preset the tip heights that will always be able to mix at the same height and aspirate liquid from the bottom of the well across all 96 channels. Additionally, with MicroPro pipetting, perpendicular and maintaining a steady pace will be maintained throughout the entire protocol. We would like to conclude this seminar by summarizing and comparing all instruments that can be used for bead cleanup. The selection of the best instrument will depend on various factors. Keep in mind, manual systems offer great flexibility, little training, except for good pipetting techniques, and always lower cost. However, using pipettes will bring higher viability across all samples, and they can limit the throughput. 96-channel systems, such as MicroPro, can be an excellent tool for this protocol and many other assays, since it's more effective or cost-effective and can be used for many workflows, including PCR setup, NGS library preparation, and more. We really appreciate you joining us today. As a reminder, the screen shows you QR codes in case you want to learn more about Rainin products like MicroPro, and there's also a downloadable case study featuring a genomics core facility at the University of Michigan and how they incorporated MicroPro into the NGS workflows. We also want to remind you that our teams will be glad to discuss more about pipetting techniques, the ergonomics aspects, and other relevant topics during our Good Pipetting Practice seminars.
Thanks for joining us today, and I hope you found something useful.
We'll see you next time.